Okay, we are now videotaping the fourth week on this molecular endocrinology lecture on the posterior pituitary hormones and POMC. And we have put all these hormones together because they would control blood pressure, blood volume, osmolarity, and so on. Basically, the posterior pituitary hormones are from the pituitary gland of the posterior pituitary neurons. It's also having the effects on the kidney from the ADH, antidiuretic hormone. Sometimes it's known as vasopressin or adrenaline vasopressin. And the second hormone is oxytocin that will control the mammary gland and also the uterine muscles for muscle contractions. At the same time, the POMC from the anterior pituitary gland would also be related to blood pressure, so therefore we put them all together here. And then there are 3 lectures or 3 videos here. Video 1 will cover posterior pituitary hormones, oxytocin and its actions. And then video 2 will focus on vasopressins and the receptors for oxytocin and vasopressin. And then in video 3, we'll talk about the evolution of these hormones known as neurohypophysis hormone from oxytocin to urotensin. We'll also mention about renin angiotensin system here. And finally, video 3 will cover POMC to end up with some exercise for these 3 videos or this part of the teaching on posterior pituitary hormones and POMC. So let's start with this video 1 with the following slides, starting from the structure of the posterior pituitary gland. Well, the hormones coming down from the hypothalamus. Here we have the PVN and SON, the supraoptic nuclei, and paramentical nuclei, together with the optical chiasm here, controlling the hypothalamic nerve. We call this hypothalamic hypophyseal area. So these are neurons producing this hormone and going down at the posterior loop. So we call this part the neurohypophysis because these are the neurons coming all the way down here. And then the artery would come in from the inferior hypophyseal artery going in. They form a network of these blood vessels going out as efferent vein going out to release the 2 hormones. So these are the hormones oxytocin and ADH, antidiuretic hormone, or also known as vasopressin. The only differences between these 2 hormones are on these 2 locations with isoleucine and also phenylalanine. And then there's a leucine in place of adrenaline here for vasopressin. So sometimes they call it adrenaline vasopressin. And then you can see that the C terminal are also amidated. So this is the first amino acid. So that is the N terminal for this neuropeptide. Okay, and then because they have 2 cysteine here, therefore disulfide bond would form here to form the secondary structure of these 2 neuropeptides. For the POMC, we'll talk about this in video 3. Actually, it would come out as ACTH in the anterior loop. POMC stands for opiomelanocortin. It is a polypeptide of 267 amino acids. And then by cutting off this into different peptides, one of the dominant one from the anterior pituitary loop is this ACTH. We'll come back to that later. As mentioned, adrenaline vasopressin has this structure with the disulfide loop. So you can see that the side chain of this phenylalanine and also isoleucine would make the foundation difference of the amino acids or the neuropeptide that would act on the perspective receptors. But what I'm trying to explain to you is that because they are actually highly similar despite of this two amino acid difference, so perhaps they have the same origin. As a matter of fact, from the cloned genes of these two hormones, we identify the structure of the genes. They all have the same structure with exon 1, 2, and 3, exon 1, 2, and 3. Number 2, they are linked together but at opposite orientation. So oxytocin gene would be forming from 1 and 2 and 3 like this, and then vasopressin gene would be forming from exon 1, 2, and 3 like this. The dual promoters actually is in between the two genes, and then you can see that the gray part here would control vasopressin, and then this open circle would control oxytocin. There are also other composite hormone response elements which look like for the control of ERE or estrogen receptor response element. These are highly conserved and found in different animals. So these regulatory elements are also for steroid factor 1 or COOP transcription factor. COOP means chicken or albumin upstream promoter transcription factor. So these elements would open up the two genes, but they are actually under different promoter to control the expression of these two genes. Now the gene products from the exon 1 to 3 actually would form into a polypeptide like this of around 90-something amino acid in length. The signal peptide is here, and then adding this together, this is the peptide to be cleaved off of around 95 amino acid. The first 9 amino acids belong to the hormone itself. In this case, it's oxytocin. And also there are also cleavage sites that would release this peptide. And this is the larger peptide, also known as neurophysin. So we take a better look of these two hormones from their pre-pol AVP or pre-pol arginine vasopressin and also pre-pol oxytocin. From the signal peptide, we guide these peptides into the rough ER, and then the dipeptide peptidase would remove the mature hormone from the precursor. The precursor would contain another peptide which is also important keeping the function of these two hormones. These are the neurophysin 2 in pre-pol arginine vasopressin or neurophysin 1 for oxytocin. And then there's an extra glycopeptide longer than this normal neurophysin 1. This is the basic structures of these two hormones. Okay, now we move on to the actions of these oxytocin. So the oxytocin in Chinese is called 催乳素, or sorry, it should be 催产素. Okay, 催乳素 is prolactin, so don't mix them up. In this case, for oxytocin, 催产素, it would also promote the secretion or release of milk. Okay, so therefore sometimes the Chinese name could be confusing. For oxytocin, after suckling, that would be the signal going over to stimulate the hypothalamus to release the hormone in the posterior pituitary gland, or also known as neurohypophysis, because it's all neurons go all the way down to the mammary gland and also to the uterus. So the uterine muscle contraction would bring about, and that would be the action for the delivery of the baby, or sometimes we call this labor. And this would bring about the distension of the cervix, with also together with suckling would stimulate this hormone. And then more hormone actions, more of this action from the mammary gland and also the uterus will stimulate the release of this oxytocin. But of course, at very high level of oxytocin, the inhibition loop would go back to inhibit the release of oxytocin. So the function of oxytocin include the followings. mainly for the females, act on smooth muscle of uterus and also the myoepithelial cells over on the duct of the mammary gland. So they have a similar mechanism for muscle contractions or cell contraction, for milk release from the mammary gland, and also for the smooth muscle to contract to deliver baby. And then, so the signal is from the nipple, as I've just mentioned, and you may, if you want to understand those more, you may click into this YouTube, but I'm not going to click on the YouTube again, and you may find these videos on the Blackboard site. So this is also related to the maternity actions in general in female. One would also ask, what is the function of this hormone in men, because it's also known to function in male, mainly on social behavior. Alright, so we are not going to get into the details because this is an area of research in recent 10 years. Okay, so we'll come back to this later. The action of oxytocin from stimulation or stimuli or suckling from the sensory receptor in the breast or vascular system. So these are actually under the control of cholinergic nerve for excitation, and also antidiuretic neurons. It would inhibit the OT or arginine vasopressin release. And then for control of milk release or uterine contraction during labor, it could be used to induce labor. But if you think about this action, its natural role on onset of true labor is not confirmed yet. What we are trying to understand is that could we really use oxytocin to induce labor? The answer is perhaps no. So the release of the milk or release of the not really release of the baby, but for the labor of the baby, it requires muscle contraction, or we call this in the uterine contraction. So basically the hormone is only inducing or acting on this uterine contraction. Okay, it's not really initiating the labor contraction. Okay, so you have to be very careful about this physiological response. And then at the end, oxytocin would also induce prolactin, and also together with prolactin, they act on many maternal or mother's behaviors. And it would also inhibit ACTH secretion and also induce sexual arousal and sperm transport in males. So oxytocin will have other functions in men as well. Okay, so oxytocin would act on its receptors like this. Is act on its receptor. It's also well known to be a G-protein coupled receptor with 7 transmembrane domain. So this G-protein would release the actions of cyclic AMP and also calcium release, and also act on this calcium channel to bring in more calcium. So the net result, you can see that of the calcium action is to act on this myosin light chain kinase to stimulate phosphorylation on the myosin. Together with the actin, it would bring about muscle contraction. And then to recycle or to remove this action, we have the myosin light chain phosphatase to bring about the myosin relax or muscle relaxation here. So to make this story more complicated, okay, these are all related to uterine contraction and also milk ejection in the myoepithelial cells of the mammary gland. But if you look into the details of this action, you will find that the excitation would also bring about protein kinase C and also adenocyclase and the increase of cyclic AMP levels. And these will bring about more actions on protein synthesis or in general other cellular responses. And for the G protein, G protein would act on not just calcium release for calmodulin to bind to the calcium, it would also for longer action to stimulate nitric oxide synthase to form nitric oxide, and this would stimulate the cyclic GMP production in the guanine cyclics. And this would bring about muscle relaxation. But more importantly is that the G protein would directly act on a quicker response of RAS and RAF and ERK1 or 2, and these pathways would lead to the release of CLOX2, which is known as cyclooxygenase 2, and this is an enzyme that would promote the formation of prostaglandins. Prostaglandins are a group of the other signaling materials to excite muscle contraction. Okay, so these are the details of these different pathways leading to the muscle contraction. So most drugs development would like to act on these other pathways instead of directly acting on the oxytocin receptor. Okay, and then we may move on. So what's the function of oxytocin in man? It's also known as man's hormone or love hormone, because it's found that it would be related to human behavior, especially on man's behavior. Now this is the secondary structure. You can see that the amino acid side chain and then the disulfide bond linkage here. So this the amino acid, only this one, and also it is the leucine here. And then this is oxytocin with act on its receptor. Of course, we are not promoting the idea of using oxytocin as a love hormone. So warning is that oxytocin is not a love hormone. It's not a drug. It was actually used a couple of years or 10 years ago that some drug company would like to develop this, what they call the cradle hormone or love hormone. Because of the inhalation of this hormone, perhaps the inhalation of oxytocin will go into the olfactory loops or the nerves, and then bring about this hormone into the hypothalamus, and then to go in. But of course, we have to aware that whether the amygdala would be affected by the hormone or the neuropeptide, or whether the neuropeptide would go into the neurohypophysis is also a critical concern. Of course, our primary concern is that how this hormone works all together to affect our other body function is also very important. And so at the end, actually more actions are related not just to the hormone itself, or human behavior is likely to be more related to their receptors. So we'll come back to that later. Okay, let's stop and then we move on to the second video. So in the first video, we have explained the structure of the posterior pituitary gland. We have explained the two hormones, vasopressins or ADH, or also called adrenaline vasopressin, together with oxytocin, and they are actually from two genes, but these two genes are highly similar with two different promoter regions to control the expression of these two hormones. And then we also discussed the actions of oxytocins, mainly on mammary gland secretion of milk and also on uterine contractions. Okay, so we stop here and then we move on to the next video.